Right, we're back with Appetite and Satiety. I did a little bit of this last year, but I think it's worth a reminder again. Some definitions here. I'm not going to read out the definitions. You can read them yourselves. Not entirely impossible. Could come up with an exam question, I guess. I really don't know. We also talked a little bit, I think, about Blundell's satiety cascade, first developed in 1987. The model involves a series of behaviour and physiological events that occur following eating and which inhibit further food consumption, which is what we're going to be talking about in the context of appetite regulating proteins. It's important for energy balance and weight maintenance. Hunger drives food intake, which triggers a series of signals eventually which terminate food intake, said to be satiation, and lead to the post-meal suppression of hunger, satiety. These homeostatic processes interact with the hedonic processes in the overall control of appetite and food intake. Hedonic processes, of course, refer to our enjoyment of food. There are 4 overlapping mechanisms: sensory, cognitive, post-ingestive and post-absorptive, corresponding to physiological processes and responses in going from the state of hunger to satiety. Body fat imposes somewhat longer-term effects associated with levels of peptides in blood, for example, leptin. The physiological process of eating tends to reduce appetite both by general feelings of fullness, such as stomach stretch, but also the impact of appetite regulating proteins such as ghrelin. And the process here is another example of a negative feedback control process. So you might want to look at the generic system there and map it out yourselves in terms of what's actually happening. This is a recent refinement of Blundell's satiety cascade, integrating recent findings. See the link paper for more background. Sensory aspects include expectations, social factors in the organoleptic properties of food, which you're all familiar with, of course, as food students. Other non-food related factors may also play a role. For example, it has been suggested that carrying out demanding cognitive tests before a meal may increase energy input without affecting perceived hunger, which is an interesting one. During a meal, ghrelin levels fall, as we'll talk about shortly, contributing to a reduced feeling of hunger. After a meal, many factors contribute to the development of satiety. These include the quantity of food, its nutrient content and its energy density. Nutrient status, most particularly for glucose and amino acids, contribute to the body's response, with several hormones being involved. Fermentation and lowering testing may also contribute to the physiological feeling of fullness, reason which which I'm sure you can imagine. Adiponectin is another one of the many hormones that influence appetite. I haven't included in detail here because in this case its involvement in regulating glucose levels and fatty acid hydrolysis is a bit unclear. It seems to have some role in energy balance as well. Okay, adipose tissue and adipocytes. Adipose tissue is commonly known as body fat. It's found all over the body. It can be found under the skin, which is described as subcutaneous fat, packed around internal organs, that's said to be visceral fat, between muscles, within bone marrow, and in breast tissue. Adipocytes are the major energy storage sites in the body, and they also have critical endocrine functions, which we have been alluding to quite a bit already. There are a number of different types. White adipocytes store energy in a single large liquid droplet and do have important endocrine functions. Note that in white adipocytes, the other cellular components are constrained to the outer edges of the cells. Brown adipocytes, which store energy in multiple small lipid droplets, are specifically used as fuel to generate body heat, that is to say thermogenesis. Some adipocytes have overlapping functions and may be referred to as bright or beige adipocytes. I'm not making it up. This summarises the responses and the consequences of both starvation and obesity. Under starvation conditions, fat stores are used up, so adipocytes shrink in overall size. Leptin is reduced in the bloodstream. The hypothalamus detects elevated leptin concentrations. That is to say, this is the trigger point concentration, via binding with neuropeptide Y5 receptor. More of that in a second. Conversely, responses to obesity involved increased fat storage, with adipocytes increasing in size. The hypothalamus detects lower leptin concentration, again trigger point concentration, by binding the melanin stimulating hormone melanocortin 4 receptor, which I'm not going to mention again, but we'll look at some of these bindings shortly. I remember reading a paper a few years ago suggesting by the time children reach adolescence, the number of their adipocytes in the body is fixed and it doesn't change, just the volume of fat stored in them, which is sort of interesting in a number of contexts. These are some of the consequences of starvation and obesity, including for completeness. We'll note, of course, that starvation is not the same as fasting. One thing I do recommend is you might want to look up technical definitions of both fasting and starvation, and also obesity, just to make sure you have these things at your fingertips if they happen to come up in an exam question, for example. Okay, ignore the full and empty balloon image. This is an otherwise good image of the stomach, the vagus nerve and the brainstem and the hypothalamus process, which is how the signals are transmitted from the stomach via the vagus nerve to the brainstem to the hypothalamus, and then there's downstream effects on that. Signals from the stomach, such as stretch and the production of hormones, are involved here. Okay, and the two hormones we're going to talk most about are leptin and ghrelin. They work in opposite ways. Ghrelin increases appetite, while leptin suppresses appetite. As I mentioned, many other proteins are potentially involved, and in fact are involved, but a bit outside the scope here. We don't have the time to do all of them, so we're going to do some key examples. So leptin. It's a hormone released by adipose tissue, that's body fat. It has a role in maintaining normal weight in a long-term person. It does this by regulating hunger by providing the sensation of satiety, that is, feeling full. It's from the Greek, as many of these things are, meaning thin or light or small, and its main role is regulation of fat stores and maintaining normal body weight. In people who have normal body weight, the extension of this to people who are obese tends to be problematic, as we'll talk about briefly in a moment. So it's quite a big protein, 145 amino acid residues. It's released, as I mentioned, by adipose cells and also enterocytes in the small intestine. That is to say, cells that line the small intestines are also heavily involved in absorption. In the intestine, leptin is involved not just in higher level control of appetite, but also in the control of absorption of macronutrients. So it was discovered fairly recently in these mice, the ob mice, which are a mutant strain of mice characterised by morbid obesity and hypophagia, that is to say they eat a lot. They are deficient in the ob gene. These mice arose at random in a colony of mice in the 1950s and are proved very valuable in the study of obesity. They are technically known as being massively obese with voracious appetites. And as usual, there's links there if you want to read more about it. But the thing which we'll notice there in the bullet points is blood leptin concentration is proportional to body fat. So the ob gene was identified in 1994, and the protein it mostly encodes for is now called leptin. When fat stores increase, leptin sends a signal to the hypothalamus that stimulates a person to eat less and expend more energy. There's no need to worry about the details of hypothalamus anatomy. Leptin signals satiety by binding to neuropeptide Y synthesized in cells in the arcuate nucleus of the hypothalamus. Now it's probably worth knowing that, perhaps possibly for the exam. This binding decreases the activity of these neuropeptide Y receptors and signals satiety. So there's neuropeptide Y. There's many other roles, as we can see from the list there, including anxiety and stress and pain perception. But its other roles are not directly relevant here. Again, it's a relatively small protein. The image includes the water molecule for its comparison in size. And there's a phrase there which I've highlighted, orexigenic. Orexigenic is a substance that promotes hunger. Another key orexigenic is ghrelin. Now, we noticed from the previous slide that binding to leptin decreases the activity of NPY receptors, so signaling satiety by reducing the amount of generation of orexigenic substances. So when leptin binds the NPY cell, it reduces the synthesis of NPY, so signaling satiety, which I've just said. It doesn't affect hunger levels and food intake from meal to meal, but acts to alter food intake and control and energy expenditure over longer periods. It helps maintain normal body weight. So it acts over a relatively long time scale compared to many other appetite regulating proteins. Leptin levels are normally measured by a blood test. White adipose tissue makes and releases leptin, as we've said. When the concentration of blood leptin is directly concentrated proportional to the amount of adipose tissue in the body, again, as we pointed out a bit earlier on, that is less body fat, less blood leptin, less leptin is circulating in the blood, and conversely, when it's higher body fat, there is more blood leptin circulating. Leptin levels increase if fat mass increases over time, and they decrease if fat mass decreases over time. But there are constraints on this, particularly with morbidly obese people. They're measured in the nanogram lane per litre range, so not a lot. Slightly higher in women due to relatively higher body fat compared to men. So consequence of high levels. I'm not sure these are specifically relevant to the module, the main exception being leptin resistance, of which more soon. The condition is a long name. High leptin levels, hypoleptin, I can't even pronounce it, you can pronounce it yourselves. When we see hyper, it usually indicates high levels, so hyperglycemia, for example. Fatty liver disease is a common condition caused by the storage of extra fat in the body. Rabson-Mendenhall syndrome is associated with a very rare genetic disorder characterized by severe insulin resistance. Hypoleptinemia, just to demonstrate I can't pronounce it. Hypoleptinemia, low leptin levels. Again, hypo indicates low levels, so hyperglycemia. Dyslipidemia is associated with imbalance of lipids, which has a range of issues associated with it, and even more impressively named hypogonadotrophic hypogonadism is associated with low sex hormone levels. So there are consequences of both low and high leptin levels, I guess mostly in people who are obese. Which brings us to leptin resistance. Many obese individuals show increased appetite and food intake, despite high blood leptin levels, which seem contradictory. They are said to show leptin resistance. That is a reduced responsiveness to leptin's appetite suppressant and weight controlling influences. Exogenous means by injection, for example, or in food, in a normal food, where when lean individuals are injected with leptin, their food intake has been observed to decrease, and there's a paper link there which talks about it. However, no such effect is seen with obese individuals, which is one of the reasons why this understanding hasn't led to drugs to modify such behaviour and such issues. Overnutrition is thought to be both the cause and effect of leptin resistance in diet-related obesity. Overnutrition, particularly a high-fat diet, may induce leptin resistance in the absence of obesity by acting on leptin-responsive neurons, and leptin resistance in obesity may maintain overnutrition. So it's very much a vicious cycle, really, I guess. We'll stop here in a moment, and there are leptin books and things out there, but leptin levels are directly related to body fat levels. It's not a nutrient. There are no foods that contain it. Lifestyles that control weight, including by food intake, can contribute, of course. Okay, I'll stop there and we'll come back for the third part in a minute.